Okay, so this week we've been messing around with seawater really. This is untreated, unfiltered, natural seawater. When we get to the hatchery process, we will be filtering all our seawater down to through a series of filter bags. But for these sort of small trials, we're just trying to get ahead of ourselves a little bit so that when we do create some larvae, we've got some of the other stages in the first 2 weeks of the growing periods that we've got our heads around a little bit. So we've basically got a holding tank, a sump. We set up a pipe with the pump running straight into the top bin. And we found the cascade was quite fast, so we added a split system which can either feed 2 tiers of bin cascades or can just split the pressure, and the pipe that goes around the outside of this blue holding tank is just splitting that water flow. We don't really want to put too much restriction on the pump flow. We do have a tap so we can control the flow rate, but if I turn the tap on, or turn the pump on, sorry, you can see how the cascade works. So seawater is pumped up from the sump. It goes through a control pressure, flow rate control tap. Half of it goes around one side and straight into the sump, and the other half goes around into the cascade. Now, the cascade is flowing quite fast, whereas the split pressure isn't, so we can just flush out any air bubbles of the split system, which gives a constant flow both through the split and through the cascade. Now, our optimum is that we don't want any of these bins overflowing, apart from the last bin, because obviously there's any larvae suspended in the bin. It will overflow into the sump, and we're trying to capture any larvae and prevent it from going through any pump or sump system, purely so we can look after it a bit better, and also so we can retain it, measure it and study it through its growth. Some will grow quicker than others, and we want the slower growing ones to be kept with each other, and the faster growing ones to be kept again with themselves. There's been a lot of work going on in the past week, both in the room itself and trying to set up these processes ahead of schedule. But so you can see little bits of plastic, which is why we're doing some flushing, and we want to get rid of any chemical residue or from the plastic making process. But you can see the flow rate's quite good into the top bin. The second bin is filling up faster and is over the top of this pipework, and the third bin is emptying quite nicely into the fourth bin. So we think we can just reduce the pressure slightly on the filler bin and let that equalise with the lower bins in the cascade. Now, a bit later on we're going to try adding a mesh to the end of the pipe, because if any larvae escapes from there to there to there to there, we're going to end up with larvae everywhere in the system, which is not something we want. We're also trying to create a cascade that gently aerates and oxygenates the larvae held in those bins, keeping both the algae and the larvae in suspension, but without over oxygenating them. Too much oxygen for the algae is certainly a bad thing, and it's probably quite a bad thing for the larvae, although we haven't got that far yet. So it's quite a nice little system. We can control how much is going through the split section, the bypass section if you like, and how much is going through the cascade just by holding the pressure off on one side or the other. We do have a tap slowing down the flow into the top bin. Right now you can see there's a little vortex at the top of this pipe, and there's a little vortex at the top of this pipe, which is really good to see. So, I mean, from this early stage experiment, I'm quite happy that neither of the first 3 bins are overflowing. The 4th bin is overflowing, so that's going to be solved by having a bit of pipe work on the exit of that bin too.